everyone. Welcome back to the SaraBioinformatics YouTube channel. I hope you are doing well. Welcome to another episode of our NGS Bioinformatics Master's course. In our previous episode, we discussed the basic concept of molecular biology and we also learned about DNA and RNA. If you are joining this course for the first time, don't worry, I am explaining everything step by step starting from the basics. I have designed this course for students and researchers from different backgrounds. Some of you may come from biology or biotechnology, while others may have a background in bioinformatics, computer science, mathematics, statistics, physics, or engineering. So, my goal is to make NGS and bioinformatics easy to understand for everyone. In today's lecture, we are going to connect our molecular biology knowledge with sequencing and next generation sequencing. We will understand the journey from gene to chromosome, chromosome to genome, and finally from sequencing to NGS data. And most importantly, we are getting closer to the actual NGS data analysis part of our course. So let's begin. Today's topic is from gene to NGS. Before we start analyzing NGS data, we need to understand where that data actually come from. Our journey today is DNA, gene, chromosome, genome, then sequencing and NGS data analysis. Then we will understand what sequencing means. After that, we will briefly compare traditional single sequencing with modern NGS. Finally, we will see how a biological sample is converted into sequencing data that we can analyze using bioinformatics. So, today's lecture is basically a bridge between molecular biology and bioinformatics. Before moving ahead, let's quickly recall what we have learned so far. In episode 1, we introduced the complete NGS data analysis course. We discussed what NGS, why it is important, and what topic we are going to cover throughout this course. Then in episode 2, we discussed molecular biology fundamentals. We learned why understanding biology is important before starting bioinformatics. In episode 3 and 4, we discuss DNA and RNA. We learn about their structure, function, differences, and their importance in sequencing. So now we have the basic foundation. In today's lecture, we are going to connect these concepts with gene, chromosome, genome, sequencing, and NGS. And after that, this lecture, we will be very close to starting our actual NGS data analysis journey. Now let's understand three very important terms: gene, chromosome, and genome. First, let's talk about gene. A gene is a specific region of DNA that contains information needed to produce a functional product such as a protein or functional RNA. Next is the chromosome. A chromosome is a highly organized structure made mainly of DNA and proteins. And finally, we have the genome. The genome means the complete genetic material of an organism. Let me give you a you have a very simple example. Let me give you a very simple example. Imagine a large book. A large book. The genome is like the complete book. A chromosome is like one chapter of that book, and a gene is like a specific instruction or recipe written inside that chapter. So remember this simple relationship. Genes are present on chromosome, and chromosome together make up the genome. This basic understanding will help us later when we study genome sequencing and genomic analysis. Now we know that DNA is a very long molecules. So you may wonder how can such a long DNA molecules fit inside the very small nucleus of a cell? The answer is DNA packaging. DNA wrap around protein called histone. This creates structure called nucleosome. These structures are further organized to form chromatin, and finally the chromatin becomes highly condensed to form a chromosome. So the basic organization is DNA to nucleosome to chromatin and chromosome. Think of it like a very long threads. If you leave the thread loose, it will occupy a lot of space. But if you carefully wind and organize it, it can fit into a small space. The cell does something similar with DNA. This organization allows the cell to store a very large amount of genetic information in a small space. Now let's talk about coding and non-coding DNA. One common question is all DNA used to make protein? The answer is no. The genome contains both coding and non-coding regions. Coding regions contains information that contributes to protein production. Non-coding regions do not directly code for proteins, but that does not mean they are useless. Many non-coding regions can have important role in gene regulation, RNA production, and other biological processes. Now let's briefly understand exons and introns. Exons are regions that remain in the mature mRNA, our mature RNA after RNA processing. Introns are removed during RNA processing, so we can think of it simply as gene to RNA processing, then intron removing, then create mRNAs. This concept will become very important later when we study RNA-seq gene expression and transcriptome analysis. So understand gene structure now will make our future RNA-seq discussion much easier. Now let's understand an important concept in bioinformatics, the reference genome. A reference genome is a representative genome sequencing that is used as a standard for comparison. When we perform NGS sequencing, we get a large number of sequencing reads. In many analysis workflow, we compare or align these reads against a reference genome. For example, imagine that we sequence DNA from a patient. We can compare the patient sequencing read with a reference genome. This can help us identify differences or genetic variations. Later in this course, when we study read alignments and variant calling, this concept will become very important. So for now, remember reference genome, a standard sequence used for comparison and analysis. Now let's understand what we mean by DNA sequence. A very simple word, sequencing means finding out the order of nucleotide in DNA. As we learned in our DNA lecture, DNA has four main bases: adenine, thymine, guanine, and cytosine. We can write a DNA sequence like ATG, C, GT, ACG. The sequencing technology help us determine the order of these bases. But why do we want to know this sequence? Because the sequence contains important biological information. By sequencing DNA, we can study genetic variation, mutations, disease-associated variants, microorganisms, genomes, evolution, and many other biological questions. The most important thing to remember is sequencing convert biological information into digital data. And once we have digital sequencing data, we can use computers and bioinformatics tools to analyze it. This is where biology and bioinformatics come together. Now let's understand where our NGS data actually come from. Everything starts with biological sample. For example, it could be blood, tissue, and microbial sample or another biological material. Depending on our research question, we extract DNA or RNA from the sample. Then we prepare the material for sequencing through a process called library preparation. After that, the prepared library is loaded onto a sequencing platform. The sequencing machine generates sequencing data. This raw sequencing data is stored in files such as FastQ files. And now our bioinformatics journey begins. We take these FastQ files and perform quality control and downstream analysis. So remember this important flow. First, sample preparation, then DNA, RNA, then library preparation, sequencing data and generate FastQ file, then start FastQ, then start bioinformatics data analysis. And this is a very important for our next episode because our next major topics we will be the FastQ file. We will learn exactly what a FastQ file is and why it is important. It is so important in NGS data analysis. Before we talk more about NGS, let's briefly understand Sanger sequencing. Sanger sequencing is a traditional sequencing technology. It is known for its high accuracy and commonly used when we want to sequence a specific or targeted region. However, it is generally more suitable for smaller scale sequencing. Now let's begin. Now let's begin look at NGS. NGS is a high throughput sequencing technology. It allows us to sequence a very large number of DNA fragments in parallel. This means that NGS can generate a huge amount of sequencing data. So, in simple words, Sanger sequencing is generally used for smaller targeted sequence, but NGS is designed for large scale and high throughput sequencing. This is why NGS has become extremely important in modern genomics and biomedical research. Now let's focus especially on NGS sequencing. NGS refers to modern high-throughput sequencing technology that can process millions of DNA fragments simultaneously. This technology has completely changed biological research with NGS. Research can study the complete genome, the exome, gene expression, cancer genome, microorganism, and many other biological systems. Some important applications include whole genome sequencing, whole exome sequencing, RNA-seq, cancer genomics, clinical genomics, and microbiome research. The biggest advantage of NGS is that it allows us to generate a very large amount of sequencing data, but generating the data is only the first step. Now we need computational method to process and interpret this huge amount of information, and that is where bioinformatics become essential. So throughout this course we will learn how to take this raw sequencing data and convert it into meaningful biological result. Thank you so much, guys. If you have any other questions, so please write in the comment box. And our next video, what is the actual raw data and what is FASTQ file, and what is structure and what is the sequencing rate, what is quality score and what is Fred score, and how do we understand a real FastQ file. So from our next episode we are moving from basic biological concept to actual NGS data analysis. And thank you so much watching Sara Bioinformatics YouTube channel. If you are learning something new from this course, don't forget to like this video, and so please don't hesitate. If you have any doubts, please write in the comment box. Thank you so much.